Ever wondered how scientists can cut and paste genes to create new DNA sequences? Welcome to the intriguing world of recombinant DNA technology. Recombinant DNA, or rDNA, is a form of artificial DNA that's created by combining two or more sequences that would not normally occur together. Picture it like a genetic jigsaw puzzle, where pieces from different puzzles are assembled together to form a completely new picture. This significant scientific breakthrough has opened up vast opportunities in the field of biotechnology, turning the once impossible into reality. But how does this all work? The principle behind rDNA technology is quite fascinating. It's all about the ability to join together DNA molecules from different species that would not naturally occur in the biological world. It's a process akin to a highly precise form of molecular cut and paste. Scientists use special proteins called enzymes to cut DNA at specific places, and then join these pieces with DNA from a different source. Imagine you're an artist, and DNA is your canvas. With a set of molecular scissors and glue, you can cut pieces from different canvases and stick them together to create a whole new artwork. That's essentially what scientists are doing with rDNA technology, but on a microscopic scale and with precision that's nothing short of extraordinary. This is not just a scientific curiosity. The implications of this technology are far-reaching. It's now possible to engineer bacteria to produce human insulin, a life-saving drug for millions of diabetics worldwide. It's also used to create genetically modified crops that are resistant to pests, reducing the need for harmful pesticides and increasing food security. Understanding this principle opens up a world of possibilities, from medical advancements to agricultural improvements. The puzzling world of recombinant DNA is a testament to the power of human ingenuity, turning the mysteries of life into tools for improving our world. Stay with us as we delve deeper into this fascinating topic in our next scene, where we will explore the art of gene cloning. Now that we have an understanding of rDNA, let's delve into gene cloning, the process that makes recombinant DNA technology possible. Imagine having the power to make an exact copy of a masterpiece painting, down to the last brushstroke. That's what gene cloning does, but instead of art, we're talking about genes, the fundamental units of heredity. It's like a biological photocopier producing multiple copies of a specific gene of interest. Why would we want to do that, you might ask? Well, having multiple copies of a gene allows scientists to study its function, to produce proteins in larger quantities, and even to modify it for therapeutic purposes. Now let's talk about the artists behind this masterpiece, the enzymes. The first are the restriction enzymes, also known as molecular scissors. They have the ability to cut DNA at specific locations, known as restriction sites. These enzymes are like the outline sketch in our art analogy, defining the boundaries of the gene we want to copy. Once our gene of interest is cut out, we need to paste it into a vector, a DNA molecule that will carry our gene into a host cell where it can be replicated. But before we can do that, we need another set of enzymes to join the DNA fragments together. Enter DNA ligase. DNA ligase acts like the glue in our art project, sealing the gaps between the DNA fragments to produce a stable, continuous DNA molecule. It's like the final brushstroke, the varnish that seals the finished artwork. In this way, gene cloning harnesses the natural abilities of these enzymes to create exact copies of genes. Just as an artist uses a brush, paint, and canvas to create a work of art, scientists use enzymes, DNA, and cells to create copies of genes. In essence, gene cloning is the gateway to genetic manipulation. Through it, we gain the power to understand, manipulate, and ultimately harness the power of genes. Like the artist with their brush, we have the tools to create, to learn, and to explore the vast potential of the genetic world. Scene script. Have you ever thought about the invisible workers behind this genetic manipulation? Meet the enzymes that act on DNA. Let's begin with restriction endonucleases. These are the molecular scissors of the DNA world. They recognize specific sequences in the DNA strand and cut at those points. Like a precise tailor, they snip the DNA strands in a predictable and reproducible manner, making them incredibly valuable when we want to splice a gene from one organism into the DNA of another. Next up, we have S1 nucleus, the cleanup crew. They nibble away at the ends of the DNA, removing single-strand overhangs. This creates blunt ends, which are easier for other enzymes to work with. It's like tidying up loose threads before sewing pieces of fabric together. Alkaline phosphatase, on the other hand, is the safety officer. It removes phosphate groups from the ends of DNA fragments, preventing them from reconnecting once they've been separated. It's like applying a brake to ensure the DNA doesn't run off course. Then we have the polymerase, the builder of the DNA world. It synthesizes new strands of DNA, using the existing strand as a template. Imagine a bricklayer carefully placing bricks in order, and you'll have a good idea of what polymerase does at the molecular level. Last but not least, we have ligase, the molecular glue. It seals the gaps between DNA fragments, creating a continuous DNA strand. It's the final touch, like a painter adding a finish to the artwork. These enzymes are the unsung heroes of recombinant DNA technology. They cut, clean, prevent reconnection, build, and seal the DNA, allowing us to manipulate genes for various applications, from medical treatments to genetically modified crops. They are the tools in the hands of scientists, enabling precise and controlled modifications to the DNA. Without these enzymes, manipulating genes would be like trying to build a house without tools. We have genes ready for manipulation and enzymes to do the job. But how do we deliver these genes into cells? Enter cloning vectors. Cloning vectors are the workhorses of recombinant DNA technology. These are DNA molecules that can carry a foreign DNA fragment into a host cell, where it can be replicated and expressed. Essentially, they're like taxis for genes, delivering them to their desired destinations. Now, these vectors are not one-size-fits-all. Different vectors are suited to different jobs, and today we're going to introduce you to a few of them. First up, we have the PUC19 and PBR322 vectors. These are plasmids, small circular pieces of DNA that naturally exist in bacteria. They're often used for cloning small DNA fragments in bacterial cells. They're like the compact cars of the vector world, nifty and efficient for small jobs. Next, we have yeast artificial chromosomes, or YAQs. These vectors are used for cloning large DNA fragments in yeast cells. You can think of them as the cargo vans of the vector world, capable of carrying a hefty load. Cosmids are another type of vector used for cloning large DNA fragments, but in bacterial cells. They're a hybrid of plasmids and phages, and you can think of them as the hybrid cars of the vector world, offering the best of both worlds. Then we have T vectors, derived from the Ti plasmid of the bacterium Agrobacterium tumefaciens. They're used for cloning in plant cells. They're like the tractors of the vector world, specialized for a specific job. Last but not least, we have shuttle vectors. These vectors are designed to shuttle back and forth between different types of host cells. They're like the versatile SUVs of the vector world, capable of handling a variety of terrains. These vectors are the unsung heroes, the taxis of the gene world, delivering genes to their desired destinations. Now that we have delivered the genes, how do we ensure they do their job? This is where expression vectors come into play. Expression vectors are a type of plasmid or circular DNA strand that are used to introduce a specific gene into a target cell. Unlike cloning vectors, which are primarily used to replicate DNA fragments, expression vectors are designed to ensure the gene is not only introduced, but also expressed within the cell. This means the gene does its job by producing the protein it codes for. Let's consider a few examples. The PGEX3X vector, for instance, is used to produce proteins with a glutathione S-transferase (GST) tag. This tag helps in identifying and isolating the protein of interest. The PPIC vectors, on the other hand, are used in yeast cells for efficient protein expression. Then we have CHO, or Chinese hamster ovary cells, which are mammalian expression vectors often used in the production of therapeutic proteins. Expression vectors ensure that our newly transported genes can carry out their functions effectively within the target cells. We've journeyed through the fascinating world of recombinant DNA technology, from understanding its principle to exploring its key players. We've dived into the concept of recombinant DNA, a technology that allows DNA from different sources to be combined, creating sequences that nature would never produce. We've also discovered the art of gene cloning, a technique that replicates DNA fragments, and the essential role of enzymes such as restriction endonucleases, S1 nuclease, alkaline phosphatase, polymerase, and ligase. We've navigated the realm of cloning vectors like PUC19, PBR322, YAC, Cosmid, T, and shuttle vector, which are used to introduce rDNA into a host organism. We've also delved into expression vectors like PGX3X, PPIC, and CHO that ensure the expression of recombinant genes within the host organism. These are just the tip of the iceberg in the vast ocean of recombinant DNA technology. Subscribe for more because you won't find me later. Until next time, keep exploring and keep questioning.